folks who've been here before, and if you're new, welcome. We do have some microphones around the room. Usually we have a lot more, but we have a large panel today, so you're going to be sharing the microphones a little bit more. We do ask, if you have a question, that you use the microphone. Because the folks at home, sometimes you've been those folks at home, can't hear you if you don't use the microphones. And like just now, without Bonnie's mic was not on because we didn't have the system on. It's a little bit harder to hear her, and then when we have the mics on, we can hear you so much more clearly. So I will be roaming around with the microphone if you're in the back of the room and you want to have a question, and we only have 2 here, and I have a roving microphone. But that's it. That's my only housekeeping notes.
That's awesome.
Now
it's working. I can. It's working. Yes. Cool. Cool.
So we got about 4 minutes before we go live. Feel free to get up, refill your plate, your cups with some libations, and we'll get started in a few minutes.
How are you? I'm terrific. How are you? Good to see you too. I was excited when you said it.
I haven't seen you before.
No more WebEx.
So cute. Food stuff. Anybody on the panel need another refreshment? Anything to drink?
Does anybody need anything? Maybe a water for Teri. She's probably in the little girl's room. I have one more question. Were we able to Zoom Jeff Julian in? Are we going to be okay? Awesome. Great. Great. Okay.
I'm not sure
earlier.
How he's, but I know Rocky's been working on it. Rocky's our magician.
Okay. Rocky's the man.
Yeah.
Okay.
We got 2 minutes, folks.
You want to go let Teri know we're missing her?
Yes, Teri. Is Teri in the little girl's room?
Yes. We're missing, she's in the potty.
Okay. Okay. Alrighty. Okay. Bang on the door. Tell her, hurry up.
You need like seat fillers, like the Oscar. You want me to sit there?
Right, exactly.
We went to Stand Up to Cancer, and they did that.
Yeah.
I went to get up to use the restroom, and somebody took my seat, and they said, "Oh, we're just going to fill it until you get back."
So your opening camera will be Shuli back there. Shuli will get. Hi, Shuli. Shuli will give you your time. We'll point to you when you need to start talking.
Okay.
She's coming.
Okay.
Here she comes.
Come on, Teri. This part is so awkward, right? Everybody's all quiet, waiting for. Right. Yeah. You know what? I can't even sing Happy Birthday, right? I have absolutely no singing voice, none. I've been told that since I was 2 years old, and it's true. I'll tell you in a minute.
Knife to the heart of a 6-year-old.
Come on, sweetheart. Oh, look at that face. Room next to Evie.
be brave if you're brave. I'll be brave, but only if you're brave. Yeah, we can both be heroes, like soldiers in their nice clothes, like soldiers in their nice clothes, together. And it could be just you and me. We'll be family. Just wait and see. So I will fight if you'll fight. Yeah, I will fight, but only if you'll fight. Oh, we can make it through this, like sailors in a tempest, like sailors in a tempest, together. And it could be just you and me. We'll be family. Just wait and see. So I'll be brave if you're brave. I'll be brave, but only if you're brave.
So welcome, everybody. It's so great to see you all here tonight, especially considering the weather that you all traversed to get here. I'm so grateful. Somebody very special is missing tonight. That would be Miss Danielle. But she woke up last night with a very, very icky flu, cold, whatever, and we all told her, as much as we know you want to be here tonight, you have to stay home, because we have other special people in the room here that don't need icky. But there's no doubt in my mind that she's dialed in, and she will be prompting me along the way, or giving Michelle orders to give to me to say this and to say that. So she will be here virtually. There's no doubt in my mind. And, you know, we so appreciate that song that was written by an amazing man. And our team got him and to come and sing his song at a special event we had. And he was so inspired, he decided to let us use his song forever for lung cancer. So next month we'll play it a little bit louder so you can hear all the words, because they're very poignant and it makes all the sense in the world. So Danielle wrote something for me to say, so I'm going to say it in her words, because she's my daughter and I love her dearly, and she loves all of you. The Adarial Lung Cancer Foundation believes the most important voices in the fight against lung cancer are the voices of our patients and their caregivers. Throughout the year, guest speakers and experts from around the world visit and share the most current and vital information and research updates. This living room is the time when patients come together to support one another and bring hope home. And I believe in that totally myself. I believe it starts with the patient and it ends with the patient. Everything we do, and we even catch ourselves every once in a while when we're having a management meeting or whatever, and we're talking about the future and different projects that come on our table. And you know, sometimes we get really excited about some things, and then we say, whoa, 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 wait a minute. Is this good for the patient? And you know what? It isn't always good for the patient. It isn't always the best thing we can do for the patient at that time. So that's how we keep our checks and balances and make sure that everything we do every day, every night, 365 days a year is what's best for the patient. Michelle, how many living rooms have we done remotely and combined here? In total? Well, since we started. I know we know the number. Danielle will dial in, or David will.
I just wrote the number out.
Okay, okay, okay.
On the road, it's been like
Okay. We're in the hundreds of living rooms since we started, right? It's an amazing number. And I don't know how many of you know that we don't just do it. What? We don't just do it here in this room all the time. We do it everywhere in the U.S. and soon going to South America and Europe and some other places to do this because, you know, I know you all think that we can do more here in the U.S. and certainly we can, but the United States is way ahead of some of the other countries in the world. And lung cancer is a global issue. It's not just an issue for the United States. So we're on the road in a big way to help everybody with lung cancer. So we have an amazing panel tonight. And, you know, as Danielle said, you know, we believe that patients are the answer to survival from the beginning all the way through the end. We need to hear from you. You need what you're experiencing. And we need to be able to pass that on as well as, you know, to other entities that are working on clinical trials and working on, you know, programs for you, for your survival. We've recently, and Amy's going to tell you a little bit more about this in a little while, but we've recently been working on clinical trials for patients. Patients are helping design the protocols. They're helping to design trials for just their needs by their genomic marker, not just in general, but by their genomic marker. So we have an illustrious panel here tonight. Don Stranathan, who's been here before, but have you been on a panel before here?
Last year, actually.
Okay.
A year ago today.
Okay. Don has kind of a very different experience with his lung cancer that he'll share. And we have Teri Kennedy, who is an EGFR patient. We've got Andy Trahan, who is an ALK patient. And I'm sure you all know Janet Freeman-Daily because she has also been here before, and she is a Ross Wonder. We also have Lisa Goldman over here, a Ross Wonder, but she doesn't like to sit up here. Right. But we may get her up here before the night is over. And we have our illustrious Amy Moore, who is our medical officer for the Adario Lung Cancer Foundation. We want to go around the room tonight like we usually do, but I'd like to go a little bit faster tonight than we normally do, because I want to take this opportunity for all of you to ask all of these folks, you know, the questions that come to mind after they kind of tell you about themselves and vice versa. And we have time for patients to dial in and ask questions of these folks as well. So if we start over here with Lisa, who just loves to talk, and she can kind of share her situation with us.
Okay. Am I supposed to look over there? My name is Lisa Goldman. I was diagnosed about 5 years ago with ROS1 positive stage 4 lung cancer. I did chemo and then switched over to Xalkori or crizotinib, and I've been on that for quite a while now, knock wood, and doing well.
Thanks, Lisa.
Hi, I'm Aimee Tom. This is my first time here. I'm really excited to be here. I've watched y'all online before. I feel very new to this. I just got diagnosed this past April, stage 4 non-small cell lung cancer, EGFR positive, and I have exon 21. And very strange to my case is that I have a very high PD-L1 status, so I had 100% PD-L1. They put me, my oncologist put me right on Tagrisso as first line, and it failed for me after 2 months. The cancer worsened and grew, and then I had some radiation to my spine because I had a spinal fracture. And then I got put on the triple therapy, chemo and immunotherapy, which I'm currently on still now. And within 2 months, I had a clear scan. I had no evidence of active disease. So I feel a great deal of whiplash, and I'm extremely grateful and also like a deer in the headlights, like what just happened and what is going to happen moving forward. I have my next PET scan the day after tomorrow, so I'm having some anxiety right now. But otherwise, I'm just trying to ride this ride this crazy wave. I got diagnosed at age 43. I'm an only parent to 2 beautiful kids, a little bit older now. So it's been a lot for my family, but I'm really happy to have found this community, and yeah, thank you.
Thank you. Effie?
Welcome. I'm glad you're here tonight. My name is Evie Schiffman. I was diagnosed in May, and in June I had my lower left lobe removed. I was very fortunate. I was diagnosed at stage 1. It was an accidental discovery. They removed the lower left lobe, and I have had my 6-month scan, and I am no evidence of disease, so I'm very fortunate.
Hi, Leslie Trahan. I'm a caregiver and wife to Andy, who's on the panel. And I won't say much more than that. I'll let him tell his story. I won't tell it for him, but I am his wife and his caregiver. And I'm super excited to be here. We've been trying to get here for almost 6 years. So
Oh, there you go.
we're from Louisiana, so we traveled far.
We guessed.
Okay.
My name is Tina, and I was diagnosed in 2010. I was EGFR positive from the get-go, but I did do chemo and radiation. And after my clear scan, after treatment, my doctor put me on Tarceva, and that was 7 years ago. So I've been on Tarceva for more than 7 years, and so far, so good.
Hi, I'm Sally, and I entered an early lung cancer detection study in 2004, and 5 years later, they found nodules. 5 years later, one grew. It was lung cancer. I had my lower right lobe removed and was staged 1A, and it will be 10 years in March.
I'm Joanne. I was diagnosed in September 2016, stage 4 adenocarcinoma, non-small cell lung cancer, EGFR exon 19. I think those are all the statistics. I was on Tarceva for 2 years, and then a trial I'm in at Natera for to use blood tests to measure circulating tumor DNA showed this last August a spike. A garden test showed that it was T790M, so I was changed to Tagrisso immediately. I have been on Tagrisso for 4 months, and my latest circulating tumor DNA went to 0. So that's where I am.
Awesome. Is it me, or is it hard to hear some of these folks?
A little bit.
A little.
Is it me?
No.
Okay, Rocky. Okay.
Hi, my name is Teresa, and I was diagnosed in July 2017. They found a 16-centimeter mass in my right lung, and I know you know Dr. Jablin, so I was able...
I know him well, intimately. He's seen me naked. Yeah.
I think he's seen me as well, because I had a right pneumonectomy in April, and so I'm apparently the only one in literature that they say I went directly to a neoadjuvant crizotinib, and it shrunk my 16 centimeter tumor to 6 centimeters, and it kept shrinking, and I had blood biopsy, so it's med exon 14, skipping and amplification. And after they did the pneumonectomy, I had it in my lymph nodes, and I had some pleural effusion. It was upgraded to stage 3A, and then they performed, he didn't want to do the surgery because it was really close to my pulmonary artery, but we convinced him, and he did do it.
Me too, yeah.
Yes, and so, and when they studied the residual tumor, there were no cancer cells. They said they've never seen that. And so I am pathologic complete response, and I'm ecstatic. And now I don't worry about it, but I tried to find other people, long-term survivors with med exon 14, and I haven't found any yet, but I hope to be one, and so.
Yeah.
Okay.
That's awesome. That's awesome.
Okay, I'm Barb Carlin. I'll try to do the really fast, short version. I'm here for Dave Carlin, who is watching from home today. Hi, Dave. Because he had surgery yesterday. Dave was diagnosed March of 2017 with stage 4. He's EGFR positive. He had a really good 15, 18 months on Tarceva. And then about the time the rash got so bad he couldn't tolerate it anymore, the drug apparently also seemed to stop working. And he developed, it started to progress in both his lymph nodes. And as of last weekend, we found out in his bones. So he had surgery yesterday because he's had really bad time with pleural effusion to the point where he's had more than 3 liters drained in the last week. So just think about that's a lot on your chest, literally. So anyway, you guys talk about being Nurse Ratchet, some of you caregivers. I got to do that today and drain his effusion. So I kind of knocked the wind out of him, and he's home recovering from that. But he will have Zometa infusion next week, which is to deal with the pain in the bones, and also I think it strengthens them as well when they have been compromised. So he's now on Tagrisso, and we are hoping that that will kick in for him.
And we're going to try to get him on some cannabis for the pain. So Dave, it's now on the record. That's what we're going to help you with.
It's legal, you can tell.
It's legal.
Totally.
I'm Ina, and I was diagnosed just a little over 4 years ago, accidental finding. So I was able to have surgery a couple of times, but hopefully they got all the cancer out of my left upper lobe and part of my lower. And at the moment, we're just watching the right side where I have several nodules, but nothing is growing. And I love this group, and thank you.
Ina is also the recipient of our gala super raffle item. She's wearing it right now. It's pretty darn sparkly and amazing.
I hardly ever take it off. Thank you for that.
That's great. Okay, yes.
My name is Joan, and I'm a graduate student at San Jose State. I was introduced actually to this program by Americans for Cures, which is a stem cell research advocacy program. So I'm hoping that from this I can learn more about lung cancer and how I can play a role for, like an advocacy. Thank you.
Awesome. We're happy to work with you.
Hello, excuse me, my name is Katerina. I'm on here on behalf of my mom, Ivica. She was diagnosed early November with stage 4 non-small cell lung cancer. She just went in for a cough, and that's how they found it. The internist just thought her cough sounded weird and decided not to wave it off and to pursue more tests. So she's had 2 rounds of chemotherapy, Alimta and carboplatin, and then also the Keytruda. And she had her first CT scan yesterday, so we'll know when she sees the oncologist Friday what it shows. And she has her 3rd round of chemo then. So we just continue to pray that, God and the medicine and doctors are doing their work.
Thank you.
Hi, I'm Rick. I was diagnosed with stage 4 adenocarcinoma back in February of 2015. I didn't have any of the genetic mutations, so I was put straight onto chemo and did 14 rounds of chemo over the course of a year. But luckily, in February of 2016, my scans were clear, and so I've been NED for coming up on 3 years now. So very grateful.
Good news.
Hi, Happy New Year. My name is Wayne. This is my wife, Rebecca, and her mother, Nellie.
How about the baby?
And Evie.
And Evie. And her New Year sweater.
In November of 2017, she was diagnosed with mucinous adenocarcinoma. She's currently metastatic, stage 4. She's gone through chemoradiation, 1 round or 4 cycles of durvalumab per the Pacific trial, and now she's on her 8th cycle of carboplatin, Alimta, and Avastin. And she discontinued her carboplatin after the 5th cycle. Currently, 5 cycles, which is pretty tough. Currently, we're trying to form our backup plan for a possible clinical trial. We're looking at a trial at Davis, one in UCSF coming up, which is really new, and also Stanford. Through my research, I found that she's her genetic testing tested KRAS G12D, which actually is a very common form of colorectal cancer. And so it's kind of maybe want to look outside the box of lung cancer and try to find things that are more suitable for that mutation. And so we're in the dark. Yeah, we're really in a bind of trying to figure out how to pick, and we're in the process of doing all our homework and getting some more advice from people as to finding the best course of treatment for the next round.
Are any of those trials looking at repurposing a drug from colon cancer in?
Right.
Yeah, they're repurposing something called regorafenib, which has been useful in metastatic colorectal cancer. The Davis trial is using a drug called trametinib, and there's a new drug from UCSF called RMC 4630.
Okay, that's good. Smart thinking.
My name is Fred Palmer. I was diagnosed with non-small cell lung cancer in October of 2013, and I'm currently on immunotherapy.
I'm Kathy Vonderporten. My mother, who's been here this summer, Chris Kuhn, she's 90 years old. You may remember her. She was here, and right after she got diagnosed with non-small cell lung cancer, she has a primary nodule, and she's not metastatic anywhere else in her system and is slow growing. But she's going to have SBRT next week. SBRT, targeted radiation. As a local treatment instead of surgery. And they think that hopefully it'll get most of it. So keep us in our thoughts. Thank you.
She's a 90-year-old pistol. She is. She's adorable.
She lives in the East Bay, and she lives in the East Bay.
Yeah, darling.
My name is Pat, and I am in the. I've had 2 needle biopsies in the past week. I don't have any results other than adenocarcinoma, and I'm here to learn the language and learn about treatments and find out what this stuff is. I have a sister who had metastatic lung cancer that went to her brain. She was supposed to only live 6 months. She made it 7 years with a lot of different treatments. I've had cancer of the appendix, so we don't go the usual routes. So I'm here to learn. Thank you.
Hello, everyone. Happy New Year. My name is Evangeline, and I was diagnosed with non-small cell lung cancer adenocarcinoma 22 months ago. As of August of last year, I was ecstatic because the doctor told me everything's dormant, but they still want to do the test on me to monitor every 3 or 4 months. So December, I went to MRI, December 10th, and for my head, because I was diagnosed stage 4 that metastasized to my brain and the bone of the skull. And then I had MRI December 10th and CAT scan December 11th. So it wasn't good news because the tumor in my brain had enlarged again from 1 centimeter to 4 centimeters. No, millimeters. So it's very small, but they're concerned because how it enlarged again. But the tumor on the left bone of the skull is stable. However, the tumor on my upper left lobe which originally was, when I was diagnosed, was 3.4 centimeters, and then it shrunk after 11 weeks of Tarceva. It went to 2.2 centimeters, and now, as of December, it enlarged to almost 4 centimeters. And so my oncologist says he believes that Tarceva is no longer working for me, so he ordered, they call Foundation Act blood test to test for the T790M, which I don't know yet what that means, but he said if
It's a good thing.
But if that turns positive, they will just automatically switch me from Tarceva to Tagrisso. However, if it turns out negative, they're considering to do a procedure, which is bronchoscopy versus a CT needle biopsy, to get some tissue samples from my lung, which causes, they want to know what's causing the increase of the tumor, and they found new nodules on my upper left and right lungs. But by God's grace, I'm still here, and I'm doing good. I'm still mobile, and I'm independent, and I'm so blessed to learn about this community, this family, which is very supportive, and I always look forward every month to be here because I feel the warmth and the hope, the confidence that everybody are fighting for, and which is very hopeful. I hope 2019 will be a great year for all of us. Thank you.
Thank you, honey.
Hi, everybody. My name is Ranlong Zhou. I was diagnosed lung cancer, squamous cell, stage 3, back in 2014, November. So I had a surgery back in the February 2015. So ever since, I've been under doing the CT scan every half year, then a year. So far, so good. It's been almost 4 years now. Things are looking good. So I'm looking forward to a 5th year. Thank you.
My name is Ken, and I'm a caregiver. My mom had, she was diagnosed April 2017 with EGFR L858R. So it's stage 4. It's in both lungs. So she's steady with her. She got started on Dacomidinib.
Okay. Now we're going to go to our illustrious panel of amazing, amazing patients, just like all of you. Someday I'm going to have you tell your whole story. He's not looking at me right now. Okay. Someday I'm going to have you tell your whole story about 2014 and the condition you were in when you walked into our living room. Okay. Remember that? He was just diagnosed, I think, that day or the day before.
The day before.
Yeah, and he was a mess. And that was 4 years ago. And, you know, I mean, this is hope at its finest. So I've already introduced everybody, but I'm going to start with Janet. I'm going to ask you all the same question. And then, I've got, maybe 7 questions here for all of you to answer in some form or fashion. And then Amy is going to talk about various different trials and studies we have going on for each one of your genomic markers. Oh, we do have Jeff up. Okay. Jeff, you look like a deer in the headlights. Okay. Yay. Okay. Jeff Julian was supposed to be here, but his flight got canceled. And he's our immunotherapy patient who's doing extremely well on IO. So Janet, I'd like to know when you were diagnosed, what you were diagnosed with, who was your medical team, any advocacy groups you've worked with, research groups, online communities, which ones, so you can help all of these amazing people here and online to get all the help they need for themselves.
And you gave me 4 or 5 hours for this, right?
No. So Janet, you have to do it really fast. Okay, really, really fast.
I was diagnosed in May 2011 with non-small cell lung cancer stage 3A. Back then, they were testing for EGFR, and I was negative, and I asked them to test for ALK, and I was negative. So I had chemo and radiation combined, and at the end of that, they did a scan, and all of the known tumors had shrunk, and they thought, well, maybe we can take out the lung and be done. And they did 15 tests in 16 days and found that the tumor had already gotten outside my chest and was now up on my collarbone. So, I was borderline stage 4. So we did more. I had learned online from patients. Inspire was the big community at that point. And I learned about genomic testing, and I got myself tested in a clinical trial for a 10-gene panel. I was negative for all of them. So I had more chemo followed by more radiation, and all the new tumors, all the old tumors had all shrunk or disappeared, but now it was in my other lung. And at that point, I happened to get, I was talking with the University of Colorado, who did my other testing, and they had a new test for a fusion called ROS1. I tested positive. I joined the clinical trial for crizotinib in November of 2012, and I have had no evidence of disease ever since.
And there's a method to my madness, you know.
Yes.
So Andy, I want you to do the same thing Janet just did, because then we're going to get more specific about each of your markers and you know what's happening there. So, Andrew Trahan,
so
New Orleans.
I was diagnosed in February of 2013. I had symptoms starting in November of 2012 with blood clots, and the doctors just never figured it out. They figured out that I had blood clots Christmas Day, but they still took, another couple of months before they could figure out that I had lung cancer. And that's largely because I was in a small town, and the doctors just don't know about genomic testing and what to look for. And so anyway, actually I didn't say that right. The doctors just aren't the best. They have too many patients, and they just push people out the door. Okay, so the genomic part was actually good, because as soon as I was diagnosed with stage 4 adenocarcinoma, they sent off for testing, and it came back positive for ALK. And so I took that for 2 years, and I had whole brain radiation along the way. So I'm not the sharpest tool in the shed anymore, but I'm still here. I've had radiation to my spine and to my pelvis and right hip as well, and still kicking. I've got 3 kids that I love and adore, and I'm so grateful that I get to spend time with them, and that my quality of life is still good all this time.
And an adorable wife.
Yeah, and a smoking hot wife.
Thank you, Andy. Thank you. Teri.
I'm Teri Kennedy, and I was diagnosed August of 2015, stage 4, and I was fortunate that as soon as I discovered a large mass in my right lung, I was sent to UC Davis, which is really close to where I live, and Insurance, which was a premier, and they did genomic testing right away. And so when I found out I was EGFR positive, deletion 19, and also UC Davis provided me information about Bonnie and ALCF. And so I was fortunate that I went to an institution that made me aware of this group, and the potential for learning a lot more about my disease and share and be able to share the experience with other patients and learn from them. So that was very fortunate. So since almost 3.5 years, I've been on many lines of treatment, but I responded really well to afatinib as a first-line EGFR treatment, but have progressed several times since. And through other patients, my doctors, and ALCF, I've learned about a lot of other treatments and been able to live a pretty nice life.
And didn't you just get engaged?
I did. I have two. I have 2 adult children who adore, and a single mom for a while. And I have a wonderful mate, Chris, and we actually got engaged on New Year's Eve.
Yay. So before we talk to you, Dawn, I'm going to give Jeff a chance. We've got Jeff Julian on Zoom, which, thank you, Rocky. We're streaming live all over the world right now, but we're also Zooming Jeff in from Southern California. And he is our amazing patient on, well, no longer on immunotherapy, but an immunotherapy patient.
So thank you for having me. I hope everything sounds okay. I'm sorry I couldn't make it. I was excited to be part of the panel. I was diagnosed in January of 2015 with stage 4 lung cancer, non-small cell. To make a longer story quick, I went through 7 doctor's visits to find the right treatment for me and to find what was going to work. Kind of went out and put it out there, so I learned about mutations pretty quick, but my first doctors didn't want to do it for me and wanted to fight against it. So we had to keep pushing, and we went from doctor's visit to doctor visit. And somewhere around the 4th one, I found a doctor that gave me my hope back, and Dr. Gitlitz, who was at USC at the time. And she was the one that really started pushing to get all of the testing done and get a full panel done instead of just the 3 or 4 that we had pushed through by then. And then I went through 3 more visits and finally found Dr. Goldman at UCLA and went on a trial for immunotherapy. I went on a first-line dual treatment of Yervoy and Opdivo, and I went on that for every 12 weeks for 2 years. My cancer has shrunk down to a nodule of about 2 millimeters in size that we monitor every 12 weeks right now, but I've just passed the 4-year mark, so we just keep going forward on that. For me, the organizations everybody knows about, I mean, it's the Dario Lung Cancer Foundation. There's a lot out there, and I get a chance to share my story quite a bit, but by personal impact of trying to help people that need help and reach out to me, or through the Dario Foundation is kind of really how I reach out and connect with amazing people like everybody in the room.
Jeff, you stopped taking Opdivo and Yervoy, correct?
Correct. About 2 years ago. So it was 2 years on, and now it's been about 2 years off.
Right. Immunotherapy. There it is, right there. And you had no other markers? No.
I have no other markers. Correct. Yes.
Exactly. Thank you. You look pretty cute on Zoom.
Thank you.
He's also a swim coach for Olympic hopefuls. And, you know, a dream came true for him when he was first diagnosed, thinking there was no future, to actually ending up being a coach at USC. UCLA.
Yes.
Where?
Yeah, USC. Yeah. I got a chance. I've since stepped back, but for the last 2 years, I got a chance to go back to my alma mater at USC and be an assistant coach there, which was amazing, but a little bit too much to try to do both. So I'm back at my club team at Rose Bowl in Pasadena and getting to live life and love life along the way.
Yeah. And you know, the lesson learned is just when you think there are no options, there's always more options. There are. Thanks, Jeff. Dawn.
Thank you.
I saved you for last because you're probably going to talk more than anybody else.
Sure.
And I wanted to tell Danielle, get better soon. Now, Danielle's your older daughter, right?
She's the older daughter.
Right?
The younger daughter's in Texas right now. Yeah.
Right.
Yeah.
I was, my name is Don Stranathan. I was diagnosed with stage 4 lung cancer in June of 2009. So I'm coming up at almost 10 years. And at the time, I was started on chemotherapy and radiation. And then I learned that I was adenocarcinoma. But back then, there weren't very many targeted drugs. I think Tarceva or Lotinib was one of the few. So I wasn't tested even for sequential sequencing. I was just given the drug, the targeted therapy, to see if it would work. And fortunately, it worked for the next 6 years. I was stable on Tarceva for over 6 years. So, I'm not ungrateful for the fact that they allowed me to just try it and see if it would work. So. And I'm a member of a number of groups. I'm involved with the EGFR resistors. I'm an honorary member because they call me an EGFR shredder, or shedder. And I've actually taken Xalkori for an NTRK mutation. In 2015, I reached out to Bonnie, and she connected with the organization Foundation Medicine. And I reached out for a comprehensive genomic sequencing and found out that I had no driver mutations, oncogenes. I had no EGFR, no ALK. I did have a 2% PD-L1 and some a fairly high 17 tumor mutation burden. So I tried 2 immunotherapies. They lasted for some time. I'm now on a triplet of Keytruda, Alimta, Carbo. I was on label for the first 11 infusions. I had some progression. I did the research, asked my oncologist if I could go off label. So now I'm back on all 3 and waiting to see if it works. If it doesn't, my next plan is possibly Impi and nivolumab or Yervoy and Opdivo. So that's my next plan B. Some of the organizations that I'm involved with, I'm an ambassador for Bonnie and Dario Foundation. I'm on Advocacy and Survivor Council for Longevity. I'm I just started a new with Foundation. I'm on their patient community panel. I'm on the survivor panel of Survive It. Kaiser, I'm a patient on the Patient Cancer Council for Kaiser Permanente, and I've been a consumer reviewer for the Department of Defense lung cancer research program for the last 5 years. So.
Busy boy. But I think the lesson learned here, from everybody speaking, is that you have to take charge of your cancer, and you have to reach out to whomever you can reach out to make sure that every step of the way you're getting the absolutely right thing for your cancer. So I have more questions. Okay. All right. So we'll start with you, Dawn. Where in your diagnosis did you hear about genomic profiling, which you already just said? And did you have testing prior to treatment or after? When did you begin to understand the importance of being tested?
Well, and I heard about comprehensive genomic sequencing from Bonnie, and that was November 2015. I understood the importance of testing immediately after I was diagnosed. And so I actually went back to the facility I was being treated at and asked if I could have that tissue tested. But the tissue there wasn't enough tissue on one biopsy, and they couldn't find the other one. So I just resolved myself to wait and see. When I did have progression, I knew testing was the first thing I wanted. And I understood also that cancer can mutate when it's being exposed to a treatment. So I thought, I'd want a new testing anyway. So, but the comprehensive sequencing was, my first one was in November of 2015. Since then, I've had like 6 different sequencing tests, either blood, liquid, or tissue.
Thank you. Teri, do you want me to read the question again, or are you okay?
I think I got it.
Okay, go.
I was very fortunate because when I was diagnosed, I knew nothing about genomic testing or what the importance of it was. And the doctor that did my biopsy said, you know, we're sending it to Foundation One, and my oncologist told me about it, and okay. So, that, again, very fortunate. So right at the time of diagnosis, I had genomic testing done. My tumor was so large, my oncologist wanted me to go ahead with starting chemo right away before I even knew the results of the genomic testing. So I did 2 rounds of chemo. Then in that time, I got the results back. It was EGFR positive. So changed over to an EGFR treatment. And since then, I've been tested many a times, both blood and tissue, as things have progressed and changed. And now I know the value of it.
And you know, it's very important when you're first diagnosed and you do start the discussion about testing, that you ask what kind of a test. Because sometimes a physician will say, we're going to send your tumor out for testing, but they may be only testing for certain markers. They might be only testing for EGFR or ALK. You know, it's really just been in the last couple of years that ROS1 has been a standard to check. And now we have many other markers. So you want to make sure that they're testing for every marker possible, including immunotherapy.
Yeah. Oh, one other thing. The time I was diagnosed, I had a blood biopsy and a tissue biopsy. I did a blood biopsy because that would come back sooner. My blood biopsy came back with no identified markers. So lesson learned there. And then, you know, a week or 2 later, the tissue biopsy came back and with a marker that I was, and a treatment that I was very responsive to. So be careful to cover all your bases at the
Yeah, and I'm glad you brought that up, Teri, because, you know, when you have a blood-based test, if there is something there, you can know and rely on the fact that it is there. Mm-hmm. If there's nothing there, you need to make sure you go back and back it up with a tissue test, because it's not always, you know, and the same thing can happen actually with a tissue test. It can come back, you know, no marker, because they haven't gotten into the core of the biopsy or what have you. So be aggressive, ask all the questions, make sure, you know, you're getting the right answers for your particular cancer. Andy?
Okay, I'm like Teri. I was diagnosed and I knew nothing about the markers, but my doctor did. And so he sent it off to just 100 miles down the road, and we got the results back really quick. It wasn't the full panel, but ALK was one of the things that he tested for. So it came back, and then I was on crizotinib. That was the very first thing I took. I didn't even have any chemo before. And as far as blood biopsies go, I mean, that I've had a couple of those and they were negative. And my cancer, the only progression has been in my central nervous system and my bones. That's it. I don't have anything that they can just go biopsy. I don't have any tumors like that. So it's been almost impossible to get into any clinical trial, and just so many things are more difficult with that situation. But I've got another blood biopsy scheduled, and we're gonna try to get a bone biopsy. My latest.
I was just going to say, you can get a biopsy in a bone.
Yeah, my latest scans showed that there was some, what they call it? Soft tissue attached to the bone.
Okay.
Or attached to the tumor that was attached to the bone.
Okay. So. Janet?
So my situation's a little different. At the time I was diagnosed, EGFR testing had become standard of care. And I was at Virginia Mason Hospital in Seattle, which it does a really good job, but they only know what's in the standard of care. So they tested for EGFR. And being a research geek, I had gone and found out about ALK testing, and I asked about that, and they hadn't planned to do it, but they did send it at my request. And they were both negative. So I had been doing more reading online with other patients, and at that time, they were just starting to try and look at other mutations that were available. There was a study called the Lung Cancer Mutation Consortium, and they were testing a 10-gene panel and trying to find how frequently these different mutations came up, and then also that was helping them guide people into clinical trials. So when I progressed, despite having curative treatment, I had a whole lot of tissue, fortunately, because they'd taken out a couple of lymph nodes. And I called to ask if I could be part of that trial, and they just finished up their funding. But the University of Colorado, despite the fact I couldn't fly out there because I had this big hollow tumor, they were worried something would happen, allowed me to enroll in the trial, and then they found a way to cover the funding through their testing lab, and they did the 10-gene panel. And I came up negative for everything, so I went on for more testing, for more treatment. But research continues, and they keep finding new markers. So I had heard from another patient online who was at Massachusetts General Hospital in a clinical trial for a new gene. It was so new, it hadn't been published, so he couldn't even tell me the name of it. But he says, you really ought to get tested for this. It's like, right, how do I do that? Then I found they published the paper January 2012, which was about 6 months after I got diagnosed, and named it ROS1. And he said, well, it tends to show up in patients like you that are relatively young, thank you, adenocarcinoma, negative for the other mutations. You might check it out. So I went to my Seattle doctor and said, I want to get tested for ROS1. And he says, what's ROS1? And I gave him the paper. And there was no way to get tested for it. The only place testing was at Massachusetts General Hospital, and you had to be their patient. So, I just kind of sat on that for a while. And then when I had finished my second treatment, or my second line of treatment, and they did the scans and found out that I'd again progressed, I had already planned to go out and visit my nephew. And then while I was visiting him, I said, gee, I'm in Denver. The University of Colorado is in Denver. And I sent an email to the head of the study to say, can I come over and say thank you for letting me be in this study? Because they didn't have to since I couldn't travel there. And they said, Dr. Bum will come meet with you in the lobby tomorrow morning. So I went
Paul
down.
Bunn, by the way, is very frequently called the godfather of lung cancer.
He started the Lung Cancer Mutation Consortium. Yeah. So he comes down to talk to me, and we talk for a while, and he says, we have 2 new tests. I said, is one of them ROS1? And he said, yes. I said, do you have any of my tissue left? And he said, yes. So I gave him permission to test, and the next week we did a bronchoscopy, but we couldn't get any more information. And the next day, I get an email from Dr. Bunn saying, you have an amazing ROS1 rearrangement. We have room in a clinical trial. And my pulmonologists in Seattle took it to the tumor board that night, and they couldn't conclude what it was for sure. So we had to wait for a month to see if anything grew before I could do anything about the clinical trial.
Wow.
And one nodule that was too small, the biopsy didn't do anything, but the other one grew by 50% in 4 weeks. And they said, go for the trial. It's either that or be on chemo for the rest of your life. I said, we're doing the trial. And my very first scan, everything was gone. So I've responded really well to crizotinib, and I'm still on it.
Thank you, Janet. That's a great story. I prepared all these questions, but I forgot to put one in here.
Did you want to get
yeah, Jeff. Well, he didn't really have a, do you have anything to add to that, Jeff?
My story is a little bit different. I didn't have a marker that they could target, but it was immunotherapy. So mine was, originally was PD-L1. I had a high expression, so that's why I got on the trial for it. Come to find out, though, and I do work closely with Foundation Medicine now, there's also a high mutational burden, so I did not have any targetable mutations. but I forget what it was, 17 or so mutations, which I remember the conversation at first was kind of disappointment of, I'm sorry, we don't have anything, and there's a lot, so it's really kind of odd. Now come to find out that it's why I've done as well as I have on immunotherapy, and it's still the same type of marker as it is. It's just not a direct mutation.
Yeah. Thank you for sharing that. That's so true and so important. For everybody on the panel, real quickly, because I've got more questions, and then we want to get to some trials. When did it dawn on you to get a second opinion? And did you ever get a second opinion? Let's start with Teri.
Oh, goodness. I think it dawned on me in the beginning, just because I'm the type that wants the more knowledge, more analysis. That's kind of my background. Let's see, I saw a second opinion, UCSF, Stanford pretty quickly, but more importantly, I think I was once I had progression and needed to decide on new treatment options, I was really active at seeking out second opinions and what other people were doing.
Andy?
We got a second opinion because we didn't like our oncologist in his bedside manner. And so we left that appointment. My dad started looking all over, and he found Ross Camage in Denver, and we hightailed it over to him, and we still communicate with him and talk to him about my scans today.
And do you think that second opinion changed the therapies you were to go on from that point on?
I wouldn't say changed, but some things I might not have done without him, I've done, and it's worked out.
That's a change.
Yeah. Well, yeah.
Okay. Janet?
Since I was deemed curable, and there was a standard of care for that, and I was at like the best hospital with a safety record and a great doctor in Seattle, I honestly didn't go after a second opinion. It wasn't until I was stage 4 and I realized nobody knows how to fix me that then I started looking into second opinions. Okay. And by getting connected to the University of Colorado, I found Ross Camage.
Thank you. Don?
I think, well, I was stable for 6 years on Tarceva, so there was really no reason to go out for a second opinion. But when I had progression in 2015, I was fortunate because as involved as I was in a lot of the advocacy groups, I was able to be set up with one of the top oncologists at Mass General. I think Bonnie set me up with a woman from UCLA. So now when I have progression, first thing I do is get a biopsy, get comprehensive sequencing. I have tests like PET, bone scans, so that I know that the cancer hasn't spread anywhere else, and then I reach out for a second opinion.
Jeff had about 20 second opinions, I think. I lost count.
I did. I'm not sure it was ever a plan not to get a second opinion and a third and a fourth. For me, very similar. My first 3 were horrible experiences. I mentioned the fourth one giving me my hope back, but that's because the first 3 took them away. When I go into a doctor's office and they look at me like I'm a foregone conclusion, and it's, well, we can do the standard of care, and yeah, you don't need the testing. We'll hope to get a few more months out of it. It wasn't enough for us, so we kept pushing, and hopefully, or luckily enough, I found a doctor that allowed me to keep pushing and allowed that hope to be there and to continue to push on. But yeah, it was a plan to have a second opinion and a third until we figured out what the best step forward was going to be and how we could create our best. I'm a coach by profession and trade, and I wanted the best team on my side I could get, including the Madario Foundation and all of the doctors. And it's not just my treating doctor, it was 4 other doctors I went back to for their opinion on which treatment to go for. It wasn't like immunotherapy as a trial was, at the time, a slam dunk by any means. And I didn't know when and if I'd get in. So it was waiting on something that I didn't know versus starting something. So, yeah, that was kind of my journey there.
It's awesome, and it's so important. It's so important to get second opinions, and not just from another doctor, from other patients, from other advocacy groups, from anybody that wants to talk to you and share their story, like you're hearing tonight and in other places. Knowledge is power. Knowledge is life-saving.
I just wanted to add one little thing to that, too. I was over a year or two into this advocacy before I learned that you might be going to an academic cancer center, but that doesn't guarantee that they specialize in lung cancer or your type of lung cancer.
That's right. That's right. That's right. And, that's why it's so important to get all the information you can get from all the people that are willing to give it to you. So that's awesome.
I think the other reason for second opinion is I have found that, especially if you're researching clinical trials, not every doctor knows about every clinical trial, and when you reach out to more professionals, then you find out about the clinical trials they are investigating or they're aware of, and so it helps with you're in that boat as well.
Yeah. When in your lung cancer treatment process did you want to become an advocate for your own treatment and others? We chose this panel for a reason, because you all have been advocates for yourselves, and you have all reached out and done, many things to make sure you're getting the right treatment. When in your diagnosis did you figure out you were going to become an advocate for yourself? And then that always leads to sharing with others. Don, we'll start with you. We haven't started with you yet, have we?
No.
We keep ending.
Well, I think the beginning.
Yeah, it's probably a good thing.
So I was diagnosed in 2009. So I had a neighbor that had pancreatic cancer in 2005. So I was his advocate and sort of caregiver. He was my next door neighbor. So I sort of started advocating almost immediately in 2009 for more information on treatment. I started with an organization, Inspire. And in 2011, I met a woman, Penny Bloom, that we started a long-distance relationship together, fell in love. She had small cell lung cancer. And that lasted for 2.5 years. And I finally brought her to my home in California to be her full-time caregiver when she couldn't care for herself. So in 2012, she ended up passing. Actually, next week will be 5 years. But in 2012, ABC had done a story on her relationship, how we had met online and that, and it sort of went viral. And because of that, I had somebody from a marketing company, public relations for Genentech, reach out to me because I'd mentioned I was on Tarceva. So I started doing media events that started opening up doors to advocacy groups. And Penny passed away in January 21st of 2014. And my last promise to her was that I would continue to advocate for more research and awareness for lung cancer. So being here tonight and being involved in a lot of these groups helps me keep that promise. So I advocate for others as much as I do for myself. So.
Yeah, true story.
I became an advocate day one as far as self-advocacy and learning and researching and getting involved with ALCF and understanding my lung cancer and what it meant. I was also an advocate because I felt the stigma right away, and hard to share your story without getting, oh, did you smoke, kind of thing, then and so that was frustrating to understand. And then I learned that anybody could get lung cancer and met all sorts of variety of patients, and that was really empowering to have a community, because I felt pretty lonely, like an island there. I didn't know anybody else, and there wasn't the support that you have with other types of cancers, diseases. So, and then about, I'd say, about 2 years into to my having cancer, I was realizing, lung cancer is a lot of subtypes, and my particular subtype, EGFR, and it was I was starting to get to know other patients and realizing that there was not a next line of treatment after you progress on the standard of care for EGFR. And I've heard about other patient groups forming and, realizing that we're a specific type that potentially can create a group that would provide knowledge for researchers, the medical community, anybody that's trying to work on treatments for this specific subtype. Why don't we band together, and as a group we're very valuable. We can provide data, we can help move along research. And so at the time I talked with another patient that I had met at the other side of the country, and she was very much more knowledgeable about social media and the access there, and I hadn't been a part of any social media group at that point. And so we decided to, and she brought in a few other people that kind of had similar thoughts in their head, and then we modeled after, which Janet's going to tell you about, but we heard about the ROS1 and what they were doing, and this is great, so we decided to form a group called the EGFR Resistors, and our mission was primarily to have a patient base of EGFR positive that were on the current line of treatment and either resisting or potentially resisting. And our group has grown to 1,000 people, and we've joined with ALCF about a year ago, once we kind of got our big boost to launch this, and so it's been very empowering. And we have a great website, egfrcancer.org, and our closed Facebook group that people are finding out, it's just kind of snowballing. People find out about it now. But the level of knowledge and the support within the group it's truly inspiring. It's not just a bunch of whining about this side effect or whatever. It's more sharing treatment and very scientific knowledge and
empowering each other.
Yeah, and it's so impressive and it's also I don't know, I can't say I've had a lot of other diseases or anything, but I find that lung cancer patients, we really want to help each other.
Yeah.
And and I find that very inspiring and empowering, and that we want the best for each other. We want to help each other. We want to learn from each other.
And at the end of the day, you feel good about yourself too, that you're not just helping yourself, you're helping other people.
Definitely.
Yeah.
When you're not exhausted.
Yeah. Yeah. I'm on your site, and I watch and listen to what the patients are talking to each other about.
Yeah.
And I'm inspired at what they know, and it's like, holy shit, I didn't know that.
Right. It's also really inspiring when people are expressing how grateful and thankful they are to have this group.
Exactly.
And you wouldn't have it without social media and without having a definitive group like this, because you wouldn't find each other.
Exactly. Exactly. We've got 49 minutes left, so I want to make sure Amy's got time to talk about the couple of studies we're working on and the one we're starting to talk to the ALCs about. But you've already talked about the patient voice. The patient voice is the most important thing happening right now in lung cancer. And it's leading the way to other cancers as well, because I don't think a phenomena like this has happened in another disease other than childhood diseases. Childhood diseases, the parents jump in and they won't take no for an answer, and they're all over it then, and they have no problem, yelling at physicians and demanding, you can do that for your children. It's simple. Physicians aren't as open to adults, behaving that way. But the patient voice in lung cancer has become extremely important. And I was going to ask each one of you to comment on that, but I think we already know how important that is. And I've got a couple of other questions. If you could answer as quickly as possible. Do you feel we're at the point that chronically managed disease is on the horizon for lung cancer?
I think yes, for at least some of them. It's not going to be 100%, but it looks like if these the research keeps popping pills and medicines out, then it can be a possibility.
I tell people this all the time this year and last year. The FDA has approved more drugs for lung cancer in the last 2 years than the last 5 decades. That's huge.
Wow.
that means they're listening. That means they're working.
I would say that, again, as Andy said, for some patients it's true, but there are still several patients, categories, different types of lung cancer for which we don't have an effective treatment. So I hesitate to put it in the category of chronically managed disease. There's a few of us in that category, but I'd like to see more.
Well, of course, but I think the researchers are now understanding that it's a possibility, because just 5 years ago, that wasn't the case. They had no idea we would be this far ahead 5 years down the road, that we could create this many new therapies for lung cancer.
I think Jack West just posted on Twitter this last week that if you count the patients who are doing well on some targeted therapies and on immunotherapy, that there's like, was it 20 to 30% of patients are now doing well long term?
Exactly.
Which is pretty amazing.
That's huge compared to where we were 5 years ago. So,
I don't think we're there yet, but I think we're getting there. And you know, I would have never in my wildest dreams thought I'd be here 10 years after being diagnosed with stage 4 lung cancer. And I think we're one step closer, because now they're mixing immunotherapies with chemotherapy. There's new vaccine trials, combination of targeted therapies. So I think we're one step closer to getting there.
what one thing would each one of you like to share with the audience that we haven't discussed already today? One, you know, some great little tidbit they can take home with them.
I think I'd like people to know there are a lot of different kinds of advocacy. Advocacy can simply be sitting with a chemo patient who needs someone to talk to, and can go all the way up to working as a research advocate with the researchers and helping design clinical trials. There's a large range. What's important is you find something you're passionate about, because it's the long haul, and it needs to be something you love, not something you're doing because you have to.
Agreed. Andy?
And I would say, don't lose hope. Don't lose hope. Keep your head up and just keep pushing along, one day at a time.
Mm-hmm. Yeah, I think if you can find your group of patients, that kind of your core that you can communicate with, it's a huge boost. I mean, for me to be able to have like a core group and I can share with them information and the knowledge we share back, it does provide so much hope. And it's like one of my first go-tos, like, this happened, what do you think, you know, or just this hug, support.
Right.
But beyond, you know, people that are in the trenches and know, I think it's so important to find others that can feel where you're at and know knowledge.
And I'd just say, be your own advocate. Every one of us on the panel here is in at least one or more groups as far as moderators, just making comments. And if you've only been diagnosed 2 or 3 months ago, you can give somebody hope that's just got the word yesterday. And so I think being your own advocate, get involved in these groups, comment. Somebody said they were newly diagnosed. Well, every one of these living rooms has been recorded. And I tell people, well, go back and start listening to some of the older recorded living rooms, because you'll learn more about what's going on in lung cancer faster than any other way.
And, you know, to Janet's point, there are many different kinds of advocacy groups out there, singular and groups themselves. You know, we can get you in touch with people that can help with your finances, that can help, you know, chat with your physician about getting payment for various different things, clinical trial help, that specialize in those various different things. So there's so much help out there if you need it, in many, many different ways. There's one more, Amy, go ahead.
I was going to say, did Jeff?
Oh, Jeff. I keep forgetting you, Jeff, because you're on Zoom.
Oh, no problem.
I never forget you.
Thank you. I would actually just echo my fellow panelists and just never give up hope. It's my message. My advocacy is a little bit different than how much time and effort the panelists put in. I try to share a story of hope and a story of inspiration if I can and impact anybody. So if I can just give that message here of keep the hope no matter what happens and no matter what you hear, keep the hope up. And live life and catch the little moments, and enjoy the little moments of life in your family and the reason why you want that hope behind you, but catch those little moments along the way.
Yeah, it's so important, and these folks too, as well as many others, speak to pharmaceutical companies, speak to researchers, speak in big groups like IASLC and AACR and all of these. And sometimes we, as a group and as a patient voice, have to bring them back from where they're going and get them back to the reality of this disease and the fact that, you know, there's patients out there waiting for you to get it right. And when you sit here with 80% of clinical trials that are failing and all of the things that are wrong, that's not what tonight is about, but that's why your voices are so, so important. When I give a presentation, I have one screen at the end of the presentation, and it's a microscope with the picture of a patient on the glass. And I always tell people when my talk is over, now when you go back to your offices and your labs and wherever you're going, remember, that's not a drop of blood and that's not a piece of tissue. That's a patient that wants to live. And that's what you're here for. And we're here to help you get there. Whatever we can do to help that process, we're here to do that. So now, Amy, you've got 40 minutes, that's pretty good, to kind of talk about the ROS1 study we're doing and the EGFR study and you know, what we're working on for ALCF.
Well, thank you again, everybody, for being here tonight. And I think, you know, we want to back up just a little bit and give some context for the organization and Bonnie's, you know, her vision in creating the foundation back in 2006, and then realizing, you know, a couple of years into it that there was more that we could do, and that research really held the key to all of this for patients. And so out of that, you know, they convened kind of the key thought leaders in the lung cancer space, and the Adaria Lung Cancer Medical Institute was born in 2008 to help propel innovative research that really could do what we're seeing today, help patients live longer. And, you know, that started very kind of basic initially. It was giving researchers access to patient specimens, because we know that is often a rate-limiting step in advancing research, is having access to those specimens, particularly at that point, stage 4 specimens. And then, you know, we began to grow that a little bit more to look at the possibility that certain forms of lung cancer were hereditary. So there was a study that grew out of that called INHERIT to say, can we identify patients who might be more at risk for a hereditary form of lung cancer? And then as Bonnie's, you know, visibility in the lung cancer community grew, she was approached by patients who were young dealing with lung cancer. And, you know, this was something that for doctors was an anomaly. You know, we shouldn't see 20-year-olds walking in the door with lung cancer, and they went through this, you know, doctor to doctor kind of bouncing around, being misdiagnosed, mistreated, because who suspects that an otherwise healthy young person might have lung cancer? So we started digging into that, looking at what are the genomics of young lung cancer, and found that these patients, typically under the age of 40, who are healthy, maybe, you know, have you know, background as an athlete or a marathon runner, that they had a higher rate of actionable driver mutations than the general public. So there was something uniquely different about lung cancer in these young patients. And so from that, we're beginning to query what are the reasons for that? Why would this happen in these young patients? And can we identify the exposures or the risks that may be accounting for this kind of uniquely different lung cancer in young people? And so as word of that kind of got out in the lung cancer community, then the ROS1ders heard about you know, the work we were doing in the young lung cancer space. So they approached Bonnie and they said, help us. You know, we want to do research. We need to figure out what's going on with ROS1 cancer. You know, lung cancer wasn't just one big pie anymore. We were beginning to understand that there were these driver mutations, but they still accounted for, you know, 1-2%, you know, just a small fraction of that entire picture. So Ross Wonders came to us, and we worked closely with them to help them figure out where were the needs and the opportunities and the gaps in that space, working with Janet, working with Lisa to say, what can we do? And so from that, we identified a couple of opportunities, working with investigators at Vanderbilt University, Dr. Christine Lovely, with Bob Doble at the University of Colorado, to develop model systems. Again, we need ways that we can study these tumors beyond just working with patients. So we were able to capture some of the tissues that were left over when these patients would come in for procedures, for biopsies, and any tissue that was left over after all the treatments and all the important things were taken care of, we said, we can take that and make it be of good use. So we could create, for example, what we call patient-derived xenograft mouse models. We can take a piece of the patient's tissue and plant it into a mouse that has a compromised immune system. And all that means is that we can more closely mimic a patient's cancer in a mouse model. And by doing that, we can begin to, look at how does that tumor grow in this model? How, what are drugs that might be good candidates to fight that? And so it really helps us ask questions that we couldn't ask otherwise. And then we similarly did that in the context of cell lines. Can we create patient-derived cell lines by taking a piece of tissue and growing it in plates, in an incubator, in special media? And that mimics some of the hallmarks of that patient's cancer. And that's particularly useful for discovery of new drugs, for example. And so, again, working closely with the patients, this organization was created as a family foundation, and we don't take that lightly. This room is a family, and we have earned your trust. We value that, and we view you as, what would I want for my sister, my mom, my dad? And so every decision I think that we think about comes back to what Bonnie said at the beginning: is it good for the patient? And so, we were able to help the Ross Wonders come together and settle on these projects. And today, we're still looking to get that word out there. And we would say to ROS1 patients in the community, if you know that you're ROS1, you have a procedure coming up, we have a hotline you can call, and then we'll make that information available. But call Nurse Alicia and get the information you need. We'd love to enroll you in this study and help you be an active participant in advancing research into ROS1 cancer. Similarly, for the EGFR patients, we were approached by them, as Teri explained not too long ago, and we did a similar process where we engaged with the key thought leaders in this lung cancer space, linked up with Dr. Paziane at Dana-Farber Cancer Institute, and with both of these studies, with ROS1 and EGFR, we're working with a company called Champions Oncology. And again, a determination was made that we need better and more models to advance research into EGFR cancer. Again, their approach was we need PDX mouse models, and there were specific mutations among EGFR patients that Dr. Yane was interested. So patients with, example, the T790M mutation that have progressed after treatment on osimertinib, the L858R mutation, exon 19 deletion, as well as patients with HER2 or exon 20 insertion mutations for which there aren't really any good TKI options out there. So again, we're trying to collect patients' samples for these specific EGFR patients. Again, we have a hotline you can call if you're interested in learning more about that study or might be a participant for that. So, we'll make that information available. So all of this is really reflective of the power of an advocacy organization coming together with patients who are committed advocates for research and linking up with the top investigators in the field to ask, what are the gaps? How can we address those gaps? And then through the infrastructure of the Adario Lung Cancer Medical Institute, we can rapidly execute on these innovative studies. We can do these clinical trials that nobody else is doing. We have the infrastructure to deliver on those in a timely way. We have, data repositories, we have biospecimen repositories, so we have unique assets that allow us to get out there and address spaces that we feel are making a difference in moving the needle for these unique patient populations. And so now we're doing the same exercise with the ALK-positive patient population, and they, again, approached us, and they've heard about the work we're doing with the ROS1ers and with the EGFR resistors, and they're saying, what can we do for them to help advance ALK-positive research? And so right now we're going through a similar process, talking to key thought leaders around the world to say, what projects are you working on to make a difference in ALK-positive lung cancer? And so we're coming up really with a list of options of the best research that's out there so that we can have a dialogue with ALK patients and figure out what really aligns with their needs and where they are as a community. We don't make these decisions in isolation. We don't impose our will on the patients at all. We want it very much to be a collaborative process that the patients feel that they're getting what they need to move the needle forward in their respective areas. So we take that trust, to heart. We want to make sure that we are working closely, as a group, as a team, to save lives. And so that's really kind of the origin. And beyond those studies, we're doing some really innovative things. We've been talking about liquid biopsy, so we're looking to say, with a study called Liberty, can we collect blood and plasma from early stage patients and look for signs of progression maybe before it's detectable on a scan? We're doing an innovative, getting ready to launch another separate study in the ALK space. So we're also looking at some potential projects on immunotherapy with patients who either are what's called exceptional responders. They do really well on immunotherapy, Jeff, or there are some patients for whom immunotherapy accelerates disease, and we need to understand really the reasons why. So we're looking at several, additional new studies in the coming year, and I think, there's lots of opportunity. The pace is really exciting, I think, from a research perspective, and as we go to these conferences, just seeing all the success that has been achieved in World Lung in Toronto back in the fall, the energy was really palpable. I think there was, a sense in among the physicians, among the researchers, that we're beginning to make progress. There's a lot of forward momentum, and so we're really wanting to capitalize on that and continue to make advances. So I hope that kind of captures some of the essential pieces, and then we want to open it up for questions. I don't know.
One of the things we're doing with the with the xenograft models, which I think is very important, in that we're actually doing a SMART study that is going to lead to a SMART clinical trial, rather than throwing spaghetti at the wall. Right. So when these mice are embedded with patients' tumors, they now have the patient's cancer. So then Champions Oncology uses various different therapies on those mice to see what works and what doesn't work. It's kind of like last mouse standing. But if you look at this, you look at ROS1ers, the ROS1 marker, that can be found in ovarian cancer and many other cancers, actually. Hopefully we can repurpose drugs. We heard something today about a colon drug being looked at for lung cancer. Maybe we can repurpose some things and do it faster and quicker and smarter, that it can lead to a trial that can move faster. But these patients have not only helped design the protocols and design the patient consent forms with us, we've done this all together, and they're also funding the trials, which is, this is patient power. This is patients really changing the dynamics and the way things have been done in the past. Jen's trying to tell me something. Jeff?
No, Michelle.
Oh, Michelle, sorry.
Amy, can you explain what a xenograft model is?
Yes. Oh. So that's where we can take a piece of the patient's leftover specimen, maybe from a bronchoscopy, some tissue, maybe blood, but tissue primarily, or pleural effusion. We're starting to allow for that. and we can implant it into a mouse. So these are special mice, not the mouse that you would catch in your house. I mean, these are lab-bred special mice that they allow the tumor to grow in a lab setting. So they more closely mimic the hallmarks of that patient's tumor in a mouse model. So we can grow these to sufficient numbers where we can begin to ask very detailed questions. We can test drugs against, these mice, but it's a way of allowing us to take that tissue and put it into a different model system to ask various types of questions related to tumor growth, response to drugs, response of maybe like the tumor microenvironment. There are lots of different ways that we can use that.
Can I add
yes,
something
please.
to that? One reason in ROS1 that's especially important to have more of these mice is because ROS1 has, at the moment, over 20 different variants. And at the start of this, we had one mouse model. Every drug that's on the market for ROS1 was tested against one variant. And we have no idea if it works for the others.
Right.
We need to create other models so we can make sure these drugs are going to work.
Right.
Right.
And we need to get the word out to the ROS1 patients upon progression when their cancer changes. They don't have to send all of their tissue for this study, just a portion of it.
And we're not asking you to have a biopsy for the study.
Right, exactly.
We're asking you when you have a scheduled biopsy because your doctor recommends it, that if you can give us some of your extra fresh tissue or pleural effusions. Yes,
yeah, exactly.
Right.
Please contact Nurse Alicia.
Right. And that's on the website and easy to, and it's also on the ROS1 and the EGFR sites on how to get in touch with Alicia.
And the other beauty of doing this approach is that these models then become available to academic researchers out there, who are working in the space. So, it's not something that we retain exclusively. We're making them openly available.
Exactly, and that's an important part of this, Amy, because most often, work that's done in clinical trials and studies and what have you, the findings are not shared until they're published. And that's not happening here. We're not worried about being published. We're only concerned with finding therapies and passing them throughout academia to do more work.
And there's also, there are some research grants that are designed for the researcher to develop these models, and then they hold on to them and they don't share.
Exactly.
And we share.
Right.
Exactly.
The other thing that I failed to mention is the work that we're doing with our lung cancer registry and accessing, and maybe you were getting to that, so I'm sorry.
No, I was gonna say, and Amy, what about the registry?
Okay, great. So we do have a patient registry where we collect baseline data on patients, including, what diagnosis you have, your previous treatments, your current treatments. So there's a whole slew of questions. And it's a very powerful tool that we're using to advance research. So as an example, we're doing a second study through it currently looking at patient-reported outcomes on immunotherapy. We have a number of patients, ALK-positive ROS1 EGFR patients reflected in that registry, and so we're working to continue to grow it because the more patients we have, the more knowledge we have, and it really is, the patients or their caregivers can go in and complete the survey, and so we're looking to use that as an ongoing research tool and thinking of the additional types of questions that we can ask using that. And so I encourage you to check out the link, lungcancerregistry.org. It'll take you to the site where you can go online and look at that. But that is another powerful way in which patients can actively participate in research, is by giving us your data, and that helps.
Yeah, and we created this registry, and we didn't put our name on it. It's called the patient registry. And we did that on purpose because we wanted to collaborate with other entities. So the American Lung Association is a partner, the International Association in Study of Lung Cancer, SITSI, which is an immunotherapy group. And this information is made public. And the beauty of this data, your data that you've typed in and sent off into this registry, we start to see patterns. Like take the exon 20 mutation or exon 14 or whatever. you start to see how many of the patients have these submarkers. And that kind of information can actually turn into a very smart clinical trial. Again, rather than throwing spaghetti at the wall, you're actually taking data and creating studies and trials based on facts rather than guessing.
So one thing that, as you all know, lung cancer does is it really steals away your sense of control. You know, the universe seems so random, and you want to do something about your disease. And one of the best things we can do is donate our tissue to researchers and donate our data so that they can try and find out why lung cancer arises in people, regardless of what personal habits they have. Lung cancer starts somehow, and that's the only way we're going to find a test that works for everybody to detect lung cancer, regardless of what your personal history may be.
And you as patients can dial in. You can dial in, put your own data in, but then you can also check to see how many other patients are in there and what do they look like. What are their submarkers? You know, what therapies have they been on? And all of a sudden, you're loaded with questions to ask your physician. It's like, what about blah, blah, blah? You know, I see that X amount of ROS1 patients are on this particular therapy, or they're in this particular clinical trial. Is that good for me? So it loads you up information again, power, 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 power.
What were you going to say?
Oh, I was just going to say, you know, like, I think we all kind of work relatively closely together. We have kind of bi-weekly or regular ROS1 calls as a group, same with EGFR. And so, you know, Janet thinks nothing of emailing me or bouncing ideas off of me and saying, you know, we're dealing with maybe this issue in the ROS1 community, and is that something that we want to explore? Is there a way to explore that? And, you know, based on patient-reported outcomes within their group that they can monitor. And so we want to kind of be that trusted resource where the patients can come to us and say, we're experiencing this. You know, what does it mean? Is there, you know, a research study we can design to look at that? Or, you know, it might even be patient education related, you know, materials. So,
and you send me articles when you find things that I haven't necessarily seen.
Right. It's a true partnership, I think. And
exactly.
Yeah.
Exactly.
And I think these groups have a tremendous impact that it's hard to understand. Lung cancer research and treatment is evolving so fast that it's really hard to keep up with. And those of us who have the disease are very motivated to find out about these things. We just had someone last week that her mother was progressing and she was at a hospital that wasn't familiar with ROS1. And because of our group, she was able to get her mother on the drug that just got approved for insurance 2 months ago, that even a major academic cancer center had not heard of yet.
Exactly, exactly. Yeah, it's incredible.
I have a registry question. Once we've put our data in, which I did, but now I'm at a different stage. So do I go back in and update that, or?
Yes.
Okay.
Absolutely. And we want you to keep loading the data in as often as it's
just
relevant.
lets me go in and amend what I did?
Not amend, add to.
Add to?
Right.
Yeah.
Okay.
Yeah. Yeah, we don't want you to take anything out.
Oh, okay, good. I will do it.
And we're talking about ways to kind of update the registry going forward in different modules. So it might be a module around survivorship, or ones that would, you know, we want to be sensitive to issues of survey fatigue. So we don't want to just bombard patients with survey after survey, you know, unnecessarily. So there are some updates like that that may occur after, you know, progression or at certain intervals. So there would be some things that we need to know kind of on a monthly basis. There would be some that are quarterly and some that might be annual kinds of surveys. So we're thinking about what are those modules, what do they look like, what types of questions do we need to collect and want to collect going forward, but making sure that, you know, I think Janet's kind of been a definite voice to this concern is patients get bombarded with a lot of questions and a lot of surveys, and it can be exhausting. So when we do reach out, we want it to be useful, we want it to be meaningful. We don't want to just blast you with surveys just for the heck of blasting you with a survey. We want to make sure that it's the highest and best use of patients' time, that it's at the right frequency. And so we are giving careful thought to what that looks like going forward. But
But
it is a powerful tool.
We want you to use it, though, to watch what's happening with your own cancer, right? Yeah. If you're, whatever your marker may be, and it could be KRAS, it could be, everybody's got a marker of some sort. It could be immunotherapy. Watch and see what's happening with, because as people enter more data, the data will change.
And we have an open RFP right now. We had our first annual summit for our community centers of excellence in December. And we really wanted to utilize our centers of excellence infrastructure around the country. We have about 31 sites across 13 states. And, because we know that 80% of cancer care is delivered in the local community, not at major academic medical centers. So how could we better capture maybe underrepresented patient populations? We know that of the data that's in our registry currently, it skews a little bit younger. There's a higher representation of women versus men, higher education. So capturing disparate populations, capturing small cell, some of these groups that aren't necessarily the oncogene-driven patient populations, and utilizing the community cancer or the centers of excellence to bring forward proposals to how they can achieve that and how they can continue to drive quality metrics in a community setting. So there's a lot of, I think, untapped potential, from the research side of how we work with and integrate with our centers of excellence. Yeah. So that's another big thing that's on the horizon for us in 2019 is working across our Alchemy network with our centers of excellence, with the registry, integrating those elements to continue to make new advances.
And at some point, it being a resource for community physicians to actually go in and see what's happening with these particular patients as well. Because many of the oncologists at community centers are managing many cancers. And they're not necessarily an expert on lung cancer, or ovarian cancer or whatever. So it could ultimately be a great resource for them as well, because it's almost impossible to keep up with.
Mm-hmm. Yeah. So, again, I think that's the overall picture of our vision when we created, or when Bonnie and Tony and the team created the Dario Lung Cancer Medical Institute, was that we would have kind of these sister organizations that are very complementary, with the ALCF being very patient-founded, patient-focused, about education and empowering you all to, be the advocates and empowered patients in navigating your own journeys. But then creating this other research network that could develop and lead these innovative trials and research studies, and putting in place that infrastructure that allows us to execute on those rapidly, efficiently, because we know in working with these patient groups that time is of the essence. We're not here just to kind of do research for research's sake, that we need to get things that will get into patients quickly. And so every time we look at a study, we have to look at it through that lens of how rapidly can we get this into patients. And it's always, I think, a balance between, there are patients who are very, I think, generous and want to make, obviously, a lasting contribution to the lung cancer field. And then we've got patients who are at a more urgent, critical point in their own treatment process. And they're saying, what do you have for me today? I need something that's going to save my life today. And so we have to, think about those two different needs, and, putting in place studies that will do both.
Right.
So.
Right. Right. Evie.
Yes, I don't know if this is on or not, but can you hear me? When I did my little introduction, I didn't mention that I've been here actually since 2011. And the first 4 years I was here with my husband, Neil, who was stage 4, EGFR, just like I'm EGFR. And he passed in 2015. And then I'm diagnosed a couple of years later, but I'm stage 1. And as I sit here, first of all, I just think you're all magnificent. And I know that Neil would be up there with you, cheering you on, and would be out there talking. I almost feel like there needs to be an early detection advocacy group. And also that early detection doesn't just mean you're diagnosed at stage 1, like I was. early detection also means, even if you're stage 4 or stage 3 or whatever, that there is early detection of if the drug that you're on isn't working, so that there's regression or there's recurrence for people who are, currently not showing, and even though they've been, stage 3 or 4. So I know that one of the major goals, or the major goal, is to make lung cancer a chronically managed disease. And the high road to get there is early detection. And whether it's like someone like me that accidentally, incidentally, only because I lobbied that I wanted to have a CT scan because I was concerned about my risk for heart disease and stroke. And that's the only reason why I was diagnosed. And good news is I'm fine with the stroke, I'm fine with the heart disease, but you got the lung cancer. But that's the only reason that I was detected. So I think that maybe there needs to be an early detection advocacy group, and it can be from people who are at all stages.
Right. Well, Evie, I'm glad you brought that up because I had a question in here about early detection, but we were running out of time. But I'm happy to say we're going to be working on a CT scan that's used because, the early detection program for lung cancer right now is primarily for people that smoked. More and more people are getting lung cancer that never smoked. They're not being counted in this early detection program. So we're thinking that if we created an early detection CT scan for heart disease and lung cancer, we would have more people lining up to go get this scan. And we would find lung cancer patients that went in for a heart scan, and we would find heart patients that went in for a lung scan. And I think it would surpass the stigma. People wouldn't be thinking about stigma anymore. They'd be going, well, isn't this a smart thing to get my entire chest scanned to see if anything's going on in there? So we talked about that at the COE summit. And so we are going to look into that because and work with cardiovascular people and so on and so forth. And we also have like 2 or 3 trials that we're working on for early detection. So early detection, obviously, to get to 90% survival with lung cancer, we have to have early detection because we're never going to get there just with trials and surgeries and the things we have now.
But
early detection's part.
Also learning some best practices from these community centers, from our centers of excellence, where we've put in place a series of what we consider to be best practices around low-dose CT screening, multi-D care, genetic testing, a whole continuum of what correlates with improved outcomes. And as a result, we're seeing stage shifting of some of our patients from, the teens that were stage 1 to over 65% of patients in this one center who are now diagnosed at stage 1. So can we take those best practices and deploy them on a wider scale throughout our COE network and beyond? So working within the confines of what we currently have in place, and then thinking beyond that into what are some innovative new approaches to early detection. So
we when we finished our COE summit in November, was it November or December? I feel like it was yesterday. And we had 35 of these centers from around the United States really wanting to come and learn and share with each other and do what they needed to do. James, our amazing photographer and keeper of all the facts and figures, was there, and he has just finished editing 2 days of all of the talks and everything that happened at that meeting. And we'll get it edited down to where, I think, what were you saying, like 20 different, 21 videos, but smaller videos. Oh, Evie, you would have been just beside yourself there. One of the presenters has a van, and they're just driving it through, the country, giving people CT scans for early detection purposes, especially in areas where it's an unmet need. That these people would never get a CT scan, no matter what we did. Because they're up in the Appalachians, and it's just not going to happen. That was one. Larry Heifetz was there, who has virtual tumor boards with multiple hospitals that connect with his hospital so they can look at patients' scans all over the area, where you've got powerful people helping less powerful people get the job done, right? Or I shouldn't say powerful, knowledgeable. Amazing things that went on in that meeting, and we'll be sending them all out in social media, in various different forms. And of course, yes. We had an awesome patient panel. Janet was there, of course.
And so was Dawn.
And so was Dawn. It was amazing to watch this patient panel, because at the very end, there was a conversation that this panel was very adamant about, and they spoke out, and they were heard. They were heard in this environment, which was great. So,
can I say something? You addressed having an advocacy group for early detection. I think lung cancer advocacy is built slowly over time as we've had more people. The ROS1ders, EGFR resistors, ALK positive, all formed because a group of patients got together and say, we really ought to do something. Someone needs to catalyze the people who were caught with early detection to get together and figure out what they can do to make a difference.
Yeah, and this is going to be the lung cancer patients that found their cancer by accident, because they're the ones that are going, oh my God, thank God I got in that car accident, because I wouldn't know I had lung cancer if I didn't, right? Good news, bad news. Good news, no bleeding, no internal bleeding. Bad news, we found a big mass on your lung. So, yeah, and Evie's on it, trust me. She's not just dropping a little tidbit here, right? Yeah. You don't have a mic. Now you do. Katerina, I think. Katerina.
Yeah. I just wanted to add to this conversation, if there's a way to harness what just happened with Ruth Bader Ginsburg and how she found hers by accident by falling and cracking her ribs. And so while that's sort of out there in the media, is there some way that anything we can do to be like, because it's right now in the forefront of people's minds?
We have to be careful with it, because no one has yet come out and said it was lung cancer. Right. Oh, really? It was found in her lungs.
And it's malignant, but no one has called it lung cancer.
So we can't, we wanted to jump on it and we haven't been able to.
Yeah, I did an op-ed on it, I did. And, but, she has to come out and say, I had lung cancer. You should. You should write her. That would be patient to patient. That would be awesome. That would be a great place. And I just adore her. I'm going to be, going to her movie very shortly. She's my hero. Just call her.
Again, the point is well taken that cancer shouldn't be an incidental finding, and that's, again, unacceptable. And Evie's been the champion of that, yes. And will continue to push on that front.
And Taylor Duck and, so many more out there that are in that position. I'm going to be in trouble if I don't do some housekeeping. Yeah. It's 4 minutes and 26 seconds. Danielle's probably saying, tell my mother to shut up. And so we have next living room, February 19th, Understanding New Science in Early Detection and Companion Diagnostics. Dr. Paul Billings from Natera, he is amazing. He's amazing, PhD. Events, February 10th, SoCal, Jeff Julian, SoCal, San Dimas, 5K. February 24th, Peoria, 5K. March 2nd, annual Run Amok with the Duck in Gainesville, Florida. Thank you as always to Penn TV. Penn TV, you guys are amazing. And zooming in, Jeff, was, just miraculous. I'm just amazed that we're doing all this. Thank God I don't have to. The Office Bar and Grill for the food every month that they send us. All the patients in the room and all the patients online. We're nothing without you. And thank you for the support from AbbVie, Amgen, AstraZeneca, Boehringer Ingelheim, BMS, Celgene, Dignity Health, Catholic Health Initiatives, Genentech, Lilly, Medtronic, Merck, Novartis, Takeda, and Tesaro. We would be nothing without them. We'd still be kicking up some dirt. But thank you all. Now we have 3 more minutes for more questions. So actually, we can ask questions as long as everybody would like to.
Were there any online questions?
Oh, did anybody have an online question?
So there wasn't, but it was the biggest turnout. So great 2019 start.
Oh, really? Okay.
Most people we've ever had online.
Oh, that's great. That's great.
And Danielle just wants everyone to know she's been online and missed being here.
Oh, we knew she was there.
And
There's
Dawn,
no way
thank
she
you
would.
for mentioning her age.
No problem.
Bonnie, can I just mention that as of last year, Genentech and Stand Up to Cancer and a few other groups started an early detection for all kinds of cancer, and that week is the first week in December. And that just began in 2017.
Oh, okay. Okay, great.
There's somebody over there.
Yeah.
Yeah, Barney. I have a question, sort of related to the early detection. I was about to ask the panel that every kind of a cancer patient, when you were told that you got cancer, everybody seems to want to know why. Why me? Why get cancer? Why lung cancer? Sort of want to know, how did it happen to me? Now you are the survivors. I'm sure that all of us, as a survivor, you always thinking, what did I do? What really triggered that to happen? Any of you kind of figure out, or have some sort of idea, this probably is the suspect that what I did, or my lifestyle, or maybe the habit, or maybe personality, or maybe the dining habit, or anything like that, and you can share that they might be the cause. Had me not done this, probably I wouldn't be even a lung cancer patient. So that would be my question. And my second question, really important, is that whether you're a cancer patient or you're a survivor, a lot of people that they only understand the physical damage that all the treatment has done to you as a patient or as a survivor. But on the other side, the mental part, the damage, often people refer to as a PTSD. So that part, a lot of people didn't understand or ignored. Do any of the panelists experiencing any challenge on your mental life change, or how to manage the mental part of the stress afterwards as a survivor? Any kind of experience or something that you would like to share, how you manage that?
We have 2 minutes, right?
No, you have 10 seconds.
Short.
No, you have all the time you want, because we've done our job. They'll shut down, but we can keep talking.
Okay.
Tell Danielle, mission accomplished. Oh, we do have Marcella, who is Andy's little daughter, who made everybody here a bracelet.
Aww.
So we're going to be passing those out. We bracelets, and they also brought, because they're from the amazing city of New Orleans, they also brought us, what do you call these? You call these beads?
Mardi Gras.
Mardi Gras beads.
Yeah.
With the little cancer logo on. So this is not the first time Marcella has made us bracelets.
So are we saying
She's
goodbye?
been in the business for a very long time.
Yes, sweetie. Thank you.
So are we saying goodbye and good night?
She's 12 now.
Okay.
But
You
when
can
she-
sign off, Jeff. You were amazing.
when she was
All right, bye. Thank you, everybody.
Love you.
Hi, Jeff. Sorry you weren't there.
she actually raised a few $1,000s for
That's
for
amazing.
Bonnie.
Yeah, she did. She did.
By people all over the country were buying
And ordering them.
things
They were
from-
ordering them, and then they're going to stop. The poor little thing, she's in there. She was, how old was she at the time? She was like 6, I think, making all these
What?
bracelets.
Well, I know we have a question too, so.
Oh, we do have a question.
Yeah.
You know, I have a comment. You know, I think we as human beings have a tendency, when something like this happens to us, whether it's cancer or multiple sclerosis or any other kind of a disease,
We do need to talk.
to say, why me? But you know, I found it always comforting to say, why not me? You know,
You have 'em?
why not me? I don't think we have choices. I don't think anybody did anything to get lung cancer. I don't care if they smoked or ate bad food or, you know, breathed in chemicals or what. Nobody deserves it. And I don't think anybody should ever be ashamed or feel shamed. Does that make any sense?
I think there have even been attempts by investigators like at Hopkins, for example. We know that cancers are not the result of any single mutation. It's really an accumulation of mutations over time, some of which you're born with, some of which you acquire because you went out and you laid out in the sun, or you had too much to drink, or you did this, or you did that, and some of it is just random, you know. And so there was an article a few years back that said, you know, maybe it's just bad luck. And for some people, it is. I mean, it's not that there's anything you necessarily did that brought this on you, or any single thing that we can say with, you know, total confidence accounts for, you know, any cancer. It's, you know, if you look at, we talked about tumor mutation burden and the amount of mutations that any type of cancer may have. And there's really like a continuum where investigators have queried different types of cancers. And, you know, lung cancer is kind of out here on the far side with lots of mutations, dozens and dozens. And, you know, it's the challenge of the field to figure out which of those are meaningful, what we call driver mutations, which are just kind of tag-alongs. They're just there. They aren't really mean anything actionable for you or contributing anything. They're just kind of attaching themselves and riding along. I'm a mutation, am I? You're a great prop. So I don't think we should approach it as there's something I necessarily did. We know that there are certain things we can do, maintain an active, you know, life and eating a good diet and minimizing our exposure to the sun. And, you know, if we do the things we know to do, then we can decrease our risk, but it's never going to eliminate it. So
Evie Schiffman is a great example.
Right.
She obviously has never been a smoker. Her diet is, you know, amazing. She's really fussy about what you eat. You eat good food. You don't do anything risky.
I'd like to take the mic off, silly lady.
We're over, right?
Well, are we? You're still talking.
But I'm loud enough. They can all hear me.
But we're still streaming.
Oh,
Well,
I thought it went off. Everybody's seeing this now.
There's still Michelle, how many people are online? Yeah, they're still listening to you.
I was going to mention.
But if you want to be done, we can sign off.
I just wanted to mention we're doing a and June 22nd. If anybody wants to save the date, everybody's invited. We're doing a Celebrate Survivorship. I'm going to be celebrating 10 years, but we're inviting everybody to come out and celebrate survivorship, anybody that's surviving. And it'll be in Winters, California, near USC Davis. I just booked the band that Bonnie had her last two galas. The London Essentials will be performing. I've got a couple of keynote speakers, so there'll be food. We're having it catered, so everybody is invited. And we're going to be doing things all weekend, but the main event will be on Saturday. And Bonnie's going to put something on the website as far as to RSVP. So thank you.
But you also asked a question about PTSD and the mental burden of cancer and survivorship, and I think that is a very real thing. You know, people talk about, and I'm not a cancer patient at this time, but they talk about scanxiety and, you know, putting in place the psychosocial support services that patients need, not just as they're actively navigating treatment, but also beyond, because there are, you know, I think great points of anxiety once you transition, you know, from your oncologist back to kind of routine care. And so, yeah, I know these people can speak to it obviously much better than me.
Has gotten better over the years. But still, I had one particularly bad week where a friend who was 1 year ahead of me on the trial progressed. One of my close local friends I knew from my in-person support group developed brain mets all over his brain. And the lady down the hill from me in my development died of lung cancer, like all within 1 week. And I find myself sitting with my husband watching a movie, and all of a sudden I couldn't breathe. You know, it just crept up on me. And I had thought I was over it. So you know, you just learn to live with it. The other thing that's a challenge is as you get involved in lung cancer advocacy and you make friends of all these wonderful people, what they're going through affects you also, because you know that it could impact you and you know what might happen to them. So you have to learn to live with uncertainty, which is a challenge.
Exactly.
Yeah.
Yeah. Yeah.
And the effects of treatment on, you know, people talk about chemo brain, and we don't even know, I guess, like the long-term effects of you know, some of the newer immunotherapies or TKIs, and so.
And affecting your lifestyle.
Yeah.
I think it's a brutal way to find out we really have very little control over very little.
Mm-hmm. Yeah.
Right.
And well, and another thing is those of us who have had the good fortune to be on these TKIs for longer than they ever expected are starting to develop late effects that they had no idea about. Mm-hmm. And we're, you know, in my case, I don't know if it's because I happen to be over 60 or if it's because I've been on this drug. And Dr. Camage very politely said to me, he says, well, I did tell you I would keep you alive until you died of something else.
The drugs kill you.
And I think there's also new side effects because they're experimenting now with all these new combinations that have been approved where they're mixing immunotherapy and chemotherapy, and they're coming up with all kinds of new side effects. So it's constantly ongoing.
Yeah. So that's where, you know, survivorship is another big thing for us to think about, you know, and that's a huge space. But as we are seeing more long-term survivors, really trying to engage with patients to capture what are these outcomes that you're experiencing, because we need to know, because doctors, you know, that we don't have that long history yet. So that's critically important to capture that data for the benefit of everyone. Yeah. So
what am I still doing here? You know, it's just a new anxiety, right? Go ahead.
I have a question. I got one, thank you. But before I ask the question, I just wanted to say to the question about, like, what did I do wrong? Then I asked, like, why does an 11-month-old get a brain tumor? You know, we have a family friend that just happened to. So that baby wasn't long enough to have bad eating eating habits and so on. So it's just random. But my question is, and I think this is probably a question that new diagnoses have as well. We're only 2 months in, and we're just trying to adjust on so many levels. So I still haven't had a chance to sit and just kind of cram for everything. But beyond reading the Bonnie Adario Lung Cancer Foundation booklet, which is fantastic, like, what do you all recommend, you know, how to prioritize or how to jump into this world? Because I just feel like I have confetti brain. Like, there's so much I want to do at once, and then I just get paralyzed, and I don't know what to read or where to go or who to talk to. So if you guys have any advice, you know, what would you have done differently, or what do you think is the best way to approach it so you don't just get information overload? Because this is all still Greek to me. Like, I don't understand half of what everyone's talking about.
Get involved in a group.
Yeah.
Get involved in a group.
The thing,
it's better too. Like, it's so new. Like, I think everyone's that way the first 2 months. But, you know, get involved in a group. But then, as you get involved in those groups and you learn and you see from others and get the support from others, and then as time goes along, it'll become clearer. 2 months is really soon, and everything is still very much a fog.
And when you mean group, is it like a Facebook group or those websites? I mean, I don't.
There are many Facebook. If you search lung cancer support,
just find the group.
There'll be many. Even for caregivers, there's caregiver groups, there's patient groups, there's support groups for everyone. There's many groups on Facebook.
So there's not just Facebook. Also, there's Smart Patients and Inspire.com and
Health Unlocked.
Health Unlocked. There's
mentor groups out there too.
But every group has its own personality, and you learn after a while being in it. Some are better moderated than others. Some have more patients that talk about research. Some are more about emotional support. So eventually, you find one you feel comfortable with.
And I just started a new group on Facebook. It's immunotherapy, or it's chemotherapy, immunotherapy, combos, triplets, and vaccines. And the majority of the people in that group are on, are on the triplets. Your mom's on. That's the triplet that I'm on, Keynote 189. So.
That's what I was going to say, is that, you know, as you find out more about her cancer and her treatment, try to ally yourself with information or groups that are closest related to what you're doing, because I think what we found is what we've all learned is how many different kinds of lung cancer there are. It's like all across the board, but it's been really helpful for us to connect with people, not just that are EGFR like Dave is, but people who have had brain mets like he's had, people who've had pleural effusion like he's had, because those are issues that you're dealing with, and it's somebody who hasn't had pleural effusion doesn't understand what you're going through. So both based on the symptoms and the treatments and the types is.
Yeah, you're so correct. You're so correct, because, you know, it's you have different things to share at different times, and you're a caretaker, so you're going to want to reach out to people that are going through what you're going through, because it's a different thing than actually having the cancer, because you're probably struggling with, what can I do to make my mother feel better, right? Well, what you can do to make your mother feel better is for you to reach out to some people to make you feel better, so you can help her more. But you have a tendency to reach out to people for specific things, like brain mets, like CT scans, like MR, like SBRT. What is that, you know? At the time it's diagnosed, or it happens in your mom's treatment program.
I also, we didn't mention one general place to look if you're interested in research is on Twitter, lung cancer social media, hashtag LCSM. If you search for that, a lot of doctors post the most newest journal article or something that's just been demonstrated to be effective, or you might see discussions about whether or not a particular treatment should be used in a certain situation. It's a really good place to get the research and clinical stuff from the doctors, as well as hear patient viewpoints.
Yep. So, yeah, absolutely. Have you noticed I'm tweeting?
Yes, thank you. Thank you.
Whoever thought that was going to happen? Miss Technology here. I'm tweeting. I'm so proud of myself.
You can run for president now.
Well,
we're not
just about anybody can do that.
Are we still live?
Yes, I know. Right. Let's call it a wrap.
Yep. Okay.
So we can elect a new president. Did you get your
bracelet? Yes, I did. Thank you.
Great meet panel.
Oh, I'll be brave if you're brave. I'll be brave, but only if you're brave. Yeah, we can both be heroes, like soldiers in their nice clothes, like soldiers in their nice clothes, together. And it could be just you and me. We'll be family. Just wait and see. So I will fight if you'll fight. Yeah, I will fight, but only fight. Oh, we can make it through this like sailors in a tempest, like sailors in a tempest together. And it could be just you and me. We'll be family. Just wait and see. So I'll be brave if you're brave. I'll be brave, but only if you're